Good evening. I am going to open a poll just to make sure that you can hear me. Hi Rita, can you hear me? That's good. Okay, perfect. Okay, I'll just remove this and we can start. Not many people now. It always happens with the last lectures. People start giving up slowly. Okay, so today we are on lecture 8 and we are going to be talking about mass spectrometry. So what we're going to go through, our learning objectives are to discuss the principles of mass spectrometry, describe the main components of a typical instrument, and then list some of the application of mass spectrometry in the biopharmaceutical analysis. And we are moving through our table. We are now down here. So as you can see, mass spectrometry can give us a huge amount of information about our product. So it can tell us information about its purity, integrity, molecular weight, amino acid composition, isoforms, post-translational modification, carbohydrate structure. So it's an absolutely probably the most powerful method that we have at the moment. Unfortunately, method, even though it's quite powerful, it is quite complex. The instruments are really expensive. The operators need to be highly trained in order to use it. And lots of methods are not as reproducible as they would like them to be for the QC use. So mass spec is still not used as much in a QC lab, but is extensively used during characterization, and it's probably the most important technique that we have when we are characterizing our drug at the beginning of its lifetime. So what is mass spectrometry? There is quite a good definition by John B. Fenn that was involved in development of mass spectrometry. So mass spectrometry is the art of measuring atoms or molecules to determine their molecular weight. And to do that, we have to put charge on the molecule of interest and then measure how the trajectories of the ions respond in vacuum to various combination of electric and magnetic fields. So he kind of said it all. We are basically using mass spec to measure a molecular weight of a charged particle inside a vacuum when it's exposed to electric and magnetic fields, so we can change its trajectory. So we will explain all of that as we go through our lecture. So you can look at mass spectrometry as a technique for weighing molecules at an atomic level. And it's an instrumental method for identifying the chemical constitution of a substance by means of separation of gaseous ions according to their different mass and charge. And then those mass measurements we are using to identify or to verify and quantitate, so we can quantitate, measure the amount of lots of things: proteins, peptides, carbohydrates, and natural products, drug metabolites. So it's really, mass spectrometry is used in a huge amount of applications. And of course, for us, the most important thing is that we can analyze proteins and peptides and carbohydrates using mass spectrometry. So in pharmaceutical analysis, it is used to study drug metabolism, so pharmacokinetics. It is used for characterization of potential drugs, to look at drug degradation, so how is drug degrading and what are the degradation products, and for screening of drug candidates, and then identifying drug candidates. For biomolecule characterization, we are using it to characterize our proteins and peptides, so most of our biologics are proteins and peptides, but also oligonucleotides and glycans. And where it's used actually quite a lot is in clinical settings for drug testing and hemoglobin analysis. This is where it would actually be used in a day-to-day lab to measure things like hemoglobin or let's say vitamin D in clinical settings would be measured using mass spectrometry. So to be able to explain how we measure it, just to remind you that all the different elements that we have on Earth can be uniquely identified by their mass. So we have this periodic system of elements, and we know the mass of each of the elements. So different compounds that contain a mixture of these elements can also be uniquely identified by their mass. And then, of course, composition of proteins can be determined as well from their amino acid sequence all to their post-translational modification, because again, because we know mass of every different element that we can find around us. So how can we see differences in different proteins that we are looking at? So any kind of difference that we have on our protein will affect its mass. So any modification of our monoclonal antibody or any other therapeutics through addition or subtraction of some kind of functional group will change the mass of the product, and we will be able to differentiate those, to see those differences using our mass spectrometry. And those mass shifts can range from really small mass alterations to very large ones. So for example, if we have a protein, and in our protein, in some of the molecules of our protein, we happen to have asparagine deamidation, that will increase mass by 0.984 Daltons. So we will actually be able to see this difference on our mass spec. If we, for example, have a sugar, something like tetra and ternary, so quite a complex sugar like you can see here, if we have sugar added to our protein, that can increase mass to around 3,535 Daltons. So that will again give us differences in our mass spectrometry. So modern mass spectrometry is really ideally suited for characterization of complex molecules such as biotherapeutics. So how do we define mass? So we assign numerical value to the property of mass, and that is based on using carbon-12 as a reference point. So we decided to use it as a reference. So 1 unit of mass is defined as Dalton. So when we talk about mass, we talk about Daltons. And 1 Dalton is actually 1/12 the mass of a single carbon-12 atom. So a carbon that has 12 protons. So 1 carbon-12 atom then has a mass of 12 Daltons, if we are going by this mass definition. Most elements around us have more than one stable isotope. So what that means is that most of the elements around us will be present in one kind of constitution, let's say, but we can have them present in other isotopes as well. So for example, when it comes to hydrogen, 99.985% of hydrogen hydrogen atoms present on Earth have mass of 1.0078 Daltons. But there is 0.015% of hydrogen ions that actually have a mass of 2.0141. So these are two different isotopes of hydrogen. When it comes to carbon, 98.89% of carbon has a mass of 12, but there is 1.11 that has a mass of 13. Same thing with oxygen, for example, where we can see that we actually have 3 different isotopes that are different just by 1 Dalton. So 1 Dalton difference means that they just have 1 proton more. Okay, why do we care about these isotopes? We care about them because mass spectrometers are actually so sensitive that they can see isotope peaks. So they can actually separate your components depending on their isotopes as well, if the resolution of that mass spectrometer is high enough. So if an NS instrument has a resolution high enough to resolve isotopes, to actually be able to see different peaks for different isotopes, then we are saying that better mass accuracy is achieved with this mass spectrometer. Okay, so after this slide, kind of short introduction, we'll start talking about the typical components of a mass spectrometer instrument. So mass spectrometer procedures, these are kind of shortly, really short list of how the procedure goes, just to introduce you to it, and then we'll talk about each one in a little bit more details. So first thing that happens is that our sample that we are analyzing is loaded onto the mass spectrometer, and it undergoes vaporization. So our sample needs to be transformed into a gaseous phase. We want to have our sample as gas. The components of that sample then need to be ionized. So remember, we can only measure mass of charged particles using mass spectrometer. So our components need to be ionized by one of different methods that can be used. So we'll talk about different methods of ionization to form charged particles or ions. Then the ions, after they are charged, they go through a mass spectrometer and they are separated according to their mass to charge ratio or M to Z ratio in an analyzer. They are separated because we are using electromagnetic field to change their trajectories depending on their mass to charge ratio. And then the ions that are separated are detected. The ion signal is processed into mass spectra, which will show us the mass of molecule and any fragments that are derived from it. So components of mass spectrometer. So typical mass spectrometer has 4 basic parts. It has an inlet. So this is a way of introduction of sample into ion source. Then the ion source that generates ions from the sample. It has a mass analyzer that separates ions according to their masses by applying electromagnetic field, and then a detector that measures the abundance of each ion present. Mass analyzer and ion detector are under vacuum, and we will discuss why that is, just so you know. And the data output is our mass spectrum. So we are getting peaks every time we are seeing an ion present. So for the different parts, we can have many different ways of, for example, ionization, many different ways of how we're gonna insert the sample into the system, or how we're gonna analyze the system. So inlet system is usually a HPLC system. So you probably know that mass spectrometers are usually connected to HPLC. So we have HPLC-MS. So samples are first separated using HPLC, and then they undergo further separation straight into a mass spec. It can also be a CE system or capillary electrophoresis system that is coupled with mass spectrometer, or it can be a syringe pump, nanosprayed needle, or a vacuum lock. Ion sources, there are many different ones. The ones that are the most important for us are electrospray ionization, or ESI, and MALDI. So they are the ones that are used the most in biopharma. Then the mass analyzers, again, there are plenty of different mass analyzers. The one that are of interest for us in biopharma would be quadrupole, ion trap, Orbitrap, and time of flight, or TOF. And then we have detector that is giving us our mass spectrum out. As I said, ion source and mass analyzers are under high vacuum, and this is because we want ions that are formed, the ions that we ionize, we want them to go through the instrument without colliding with gaseous molecules. If we did not have vacuum in there, if we had air in there, our ions that we generated would bump into the molecules of gas that is present in there, and they would collide, and their trajectories would be changed, which means that maybe we wouldn't be able to detect them. So we want these components to be under high vacuum, so there is no air present, so there is no gaseous molecules present, and only our ionized samples are traveling through mass analyzer. Okay, so we'll talk now about ion sources that can be used. So ion sources are sources that we use to ionize our samples, so to make our samples charged, and there are many different ones. One of the oldest methods, or the first method that's been used, it's called electron ionization, or EI, or hard method. This is a method that is really good for small molecules, so molecules up to 1000 daltons. We, of course, are dealing with much larger molecules, so biopharmaceuticals are usually measured in kilodaltons. So for this, ionization methods that are much better would be electrospray ionization, which is good. It's called also soft method of ionization. It's good for ionization of peptides and proteins up to 200,000 daltons. And then we have matrix-assisted laser desorption ionization, or MALDI, which is again soft ionization, which is good for peptides, proteins, DNA, and molecules up to 500 kilodaltons. So just an example of ionization process, the way it works, how we get charged molecules. On an example of an ethane here that we have, if we start ionizing this molecule here, which is not charged at the moment, the first thing that's going to happen is when we bombard this with electrons, the different bonds in ethane will start breaking. So first thing that's going to happen is that this molecule will lose one electron. When it loses one electron, we will get a radical cation, so positively charged molecule with one unpaired electron. So this molecular ion will have mass to charge ratio of 30. So mass to charge ratio is 30 because we have two carbons, which are 12 daltons inside, so that's 24. And then we have 6 hydrogen ions, which then bring us to 30. And they have one charge. So 30 divided by 1 is 30. This radical cation, or this molecular ion, is not really stable, so it will quickly break more. And what will happen, it will lose one hydrogen, and we will have now a positively charged molecule here with mass to charge ratio of 29. And then it will also break in here, and we will get one positively charged molecule that has mass to charge ratio of 15, and another one that is not charged. So if it's not charged, this is not going to be detected by mass spec. This is just an example of ionization process. Different molecules will fragment differently when they are ionized, but usually we can predict where molecules will fragment and what kind of mass spectrum we should get. So electron ionization, so this is the hard method. This is one of the first methods that is being used. In this case, we are ionizing our samples by introducing a sample into an instrument by heating it until it evaporates. Once it's introduced into the instrument, this gas phase sample is bombarded with electrons that are coming from rhenium or tungsten filament. So sample comes in the system, and then it's bombarded by electrons. Molecule is then shattered into fragments, so similar to what I showed in the previous slide, depending on the molecule. And then those fragments are sent to mass analyzer via repeller. So if they fragment into positively charged molecules, they will then we can have a repeller that is negatively charged, and that will push them to mass analyzer. Okay, just a second. I lost my pointer. Let's see if it's going to work again. So electron ionization will break up molecules in predictable ways. So we can predict where the molecules will break because we know where the bonds are the weakest. This is suitable for small molecules. It's not suitable for proteins because this is quite a hard method, and it shatters molecules into many different fragments, which is fine for small molecules. But imagine if we have a protein that contains thousands of amino acids. If we try to bombard that with electrons, we will just get such a huge number of fragments that we would not be able to analyze them in any way. So it just breaks our protein into too many fragments to actually be able to deal with. So this method of ionization we are only using for small molecule drugs, but it's not used for biopharmaceutical analysis. So for biopharmaceutical analysis, what we can use is soft methods of ionization, such as MALDI and electrospray ionization. Electrospray ionization, unlike the one that we mentioned before, keeps molecule of interest fully intact, so it doesn't really fragment our proteins. The way it works, sample is introduced through a needle, through an injection needle as a liquid, and as it goes through the needle is charged, and the voltage that is applied across the needle puts electrical gradient on a sample, and then sample disperses into charged droplets, as you can see here. So we have now our sample inside the charged droplet. Guys, I'm really sorry, but my pointer is not working, so I'm not going to be able to show you. I don't know what the problem is with it. Yeah, really sorry about that. Hopefully this will work without it. Okay, so we get charged droplets, and our sample is within those charged droplets. Then solvent is evaporated using heat or dry gas, and as we evaporate solvent, as you can see here in the middle, the charge will jump from the droplet onto, as the droplet is being dried, the charge will jump from it onto our sample. And then opposite voltage will attract droplets to the mass analyzer entrance, and droplets will explode into ions. So, for example, if we use something like that, ESI will typically produce a number of ions with different charge states, and that will depend on number on site in which protonation or deprotonation can occur. So as you can see here, if we have a protein that is that we use electrospray ionization to ionize it, protein will actually not going to shatter into fragments, but it might gain proton or lose proton on either side or on some areas of it. So we can get single charge proteins, double charge proteins, we can get proteins with no charge, we can get proteins again with single charge but charge on the other side of the protein. Okay, so single protein can look like this. So if we put charge on a single protein using electrospray ionization, we might get multiple charge states for a single protein, depending on what are the areas where it can gain or lose proton. So in this case, we might have 1 charge, 2, 3, 4, or 5 charges on our protein. And then, of course, depending on the charges, we will see differences in mass to charge ratio and then differences on a mass spectrum. So we are able to separate these different samples. Another soft ionization would be MALDI, or matrix-assisted laser desorption ionization. So in this case, MALDI, we have a sample plate with a matrix on it, and we mix our sample with that matrix. So this little plate is kind of like size of your palm. So your sample is now mixed with the matrix, and then we use a laser beam in order to ionize the matrix. And once we ionize the matrix, our sample will get charged because it's going to be passed to it from the matrix. And then we can send our analyte or sample that we are analyzing towards the mass spectrometer. So these are the matrices. So there are usually different acids such as DHB or THAP. So the matrix consists of these crystallized molecules, and solution of the molecules is made often in a mixture of highly purified water and organic solvent. And MALDI typically produces singly charged ions. So usually your ions are just going to have 1 charge. But if you want MALDI to produce multiple charges, then you can change your matrix, you can change your laser, you can change your voltage, you can play around with it to change the charge of your sample that you're analyzing. When you compare MALDI and electrospray ionization, MALDI is usually easier to use. You only need low amount of samples and it's highly sensitive. And the good thing is it produces singly charged ions. Then you can couple with mass analyzer that is called TOF, or time of flight, that we're going to be mentioning. So you can really analyze something that is of high mass. So if you remember, we said that MALDI is good to use if you have proteins that are really large. So proteins that are around 500,000 daltons or 500 kilodaltons. Electrospray ionization gives you increased analyte information because it's usually coupled with HPLC. So you can get information from HPLC and then furthermore more information from ESI, using ESI. It's highly sensitive. It can be coupled with wider range of mass analyzers. So MALDI is usually coupled with TOF, but ESI can be coupled with many different ones. And as I said, you can have interface such as liquid chromatography, gas chromatography, or capillary electrophoresis before you couple it with ESI. But it takes longer time to analyze your sample using ESI. But the good thing is that you get little or no fragmentation during ionization. So you're not going to be getting huge amount of data that you cannot really analyze and make sense of. Okay, so those were ion sources, most commonly used. Now we're going to talk about the mass analyzer. So after we charge our ions, so after we make charged ions, how do we then separate them based on their charge, size to charge ratio? So this is what mass analyzers do. They separate ions based on their mass to charge ratio. They operate under high vacuum, remember, because we want to prevent ions from bumping into gas molecules. And key specifications for mass analyzers are their resolution, their mass accuracy, and their sensitivity. So sensitivity means how low can the instrument go? So what is the minimum amount of material needed to generate analytical data? This is normally expressed in femtomoles, and of course, the lower we can go, the better sensitivity it is, and we are wasting less sample. Mass accuracy is telling us how close to the theoretical value is the experimentally determined mass. So we can calculate, of course, theoretical value of our product because we know exactly atoms that are in there, and then through the periodic system of elements we can calculate the theoretical value. So we want to see how close is the value that is determined by a mass spectrum is to the theoretical value. So this mass accuracy is an important feature, and it's needed when searching your databases and informatic tools to determine identity of your composition. So when you get like fingerprint and then you compare it to your database to see what you actually have. This is normally reported in ppms or parts per million. And then resolution is ability to determine charge, state, and resolve mixtures. So things like how good your mass analyzer is to be able to resolve mixtures that are quite similar. So generally, the higher the resolving power of the instrument, the better the mass accuracy. They're all linked. And this is reported in resolving power, or RP. So remember, if your mass analyzer can see between isotopes, that means it has quite high resolution. So one of the first mass analyzers or original mass analyzer would be magnetic sector analyzer, or MSA, which gives you really high resolution and really exact mass. Then the ones that are used in biopharma more would be quadrupole analyzer, or Q or quad analyzer, which gives quite low resolution, but it's quite fast and cheap. Then we have time of flight analyzer, or TOF, which gives you high throughput, and you actually don't have any upper limit on your mass to charge ratio to be used for this one. So magnetic sector analyzer, this one works in a way that it generates a field at right angles to your moving ions, causing them to travel in these curved trajectories through an analyzer. So by changing the electromagnetic field, you can make certain mass to charge ratio be able to travel through the mass analyzer and hit this detector. Ions that are too light, they will change their trajectory and they will actually fly through, but they won't hit the detector. And the same will happen with the ions that are too heavy. Okay, so by changing the electromagnetic field, you're actually picking what mass to charge ratio will go through this curved trajectory and hit the detector. Okay, so by varying the field, it allows us to different masses to pass down the center and be detected. Quadrupole mass analyzer is consists of 4 parallel circular rods, as you can see here, and each opposing rod pair has a direct current or radio frequency. So we can change this direct current or radio frequency. And depending on how we change it, again, we can pick certain ions, certain mass to charge ratio to travel down the flight path. So we can just basically zoom in on some, like the one in blue that you can see here. So that is a certain mass to charge ratio that will go through quadrupole filter all the way to the detector. The purple ones or the yellow ones or the orange ones will be kind of skewed from the trajectory and they will not be detected. So only ions of certain mass to charge ratio will pass through the quadrupole filter. This is really useful when you want to study particular ion because you can stay tuned on single ion for a really long period of time, let it hit the detector pretty frequently, and then you get a lot of information from that particular ion. Time of flight, in this case we have ions flying along a really long tube, and we are measuring the time required for ions to fly down the length of the tube. So we just put them through the tube and they're flying through. Smaller ions will get to the detector first, and then they will be followed by medium and large ions. So we are basically separating them based on size. I'll just go back. So these tubes can just be kind of one tube. They can be in V shape, as you can see here, or they can even be in W shape. So if you want, you can give your sample longer time to spend within mass analyzer. And the longer time they are spending, the better the separation you will get. As you can see here, if you look at the graph, the first graph, you can see that these three colors, these three different sizes are overlapping, but if you let them travel through a longer path, they will actually separate much better. Then we have ion trap mass analyzer. So ion traps are essentially just like quadrupole, but bent around on itself. So they use a combination of DC and RF voltages to trap ions. And then, so you're kind of trapping ions, and then when you have enough, you're ejecting them in sequential manner. So this larger ion storage capacity generates better dynamic range than quads. Okay, so it kind of allows you to trap a lot and then send it straight away into the detector to be detected. So after this, we'll talk about detector and data system. So detectors are electron multipliers that produce electronic signal when struck by an ion. So when the ion goes to the detector, struck the electron multipliers, more electrons are being produced. Then they travel, they struck the multiplier, and even more electrons are being used. And by this, we get multiplying of signal. So collision of ions from our analyte or on dynode surface causes the release of more electrons, and this is what amplifies the signal, so we can actually visualize it. Efficiency of this process influences sensitivity of detection, and this detection needs constant and regular calibration. So you need to calibrate the detector before the use, so before your analysis, but sometimes even during your analysis, because they're quite sensitive. So typical mass spectrum would look something like this. So usually on the x-axis you have mass to charge ratio, and then on the y-axis you have relative abundance of certain ions. So ion abundance versus mass to charge ratio. the most abundant ion formed in ionization will give rise to the tallest peak. So this is the base peak. And then all the other peak intensities will be calculated relative to the base peak. So the tallest one, that's the one that's most abundant, we call that 100%, and then everything else, the abundance of everything else is calculated compared to this base peak as a percentage. So we have some mass definitions that are mentioned when talking about mass spec of certain elements. So we have monoisotopic mass of a molecule. That's the sum of the exact masses of the most naturally abundant isotope of each element present. So, and that would be hydrogen with 1 proton, carbon 12, or oxygen 16. So for example, monoisotopic mass of a drug, so this is a small molecule drug called atorvastatin, is 558.252991 Dalton. So this is calculated as taking into account that it only has most abundant isotopes. However, we have the average mass of the molecule also is the sum of the average masses for all elements present in that molecule. So instead of using the exact mass of most naturally abundant isotope, we're kind of using average masses. So these values represent the isotope abundant weighted average for each element. So for that, in that case, the average mass of atorvastatin will be slightly different. So it's 558.639771. So it depends whether you are reporting monoisotopic mass or the average mass. But monoisotopic mass measurements are preferred when possible because due to higher associated accuracy of your instrument. So if you're looking at the peptide with the sequence, here you can see that the most abundant peak would be monoisotopic mass, but you can see then what would be the average mass. But we would of course have some of these peptides that contain one C13 atom, and we also have some of them that contain two C13. And they can all, even though the mass between them is quite small, if your mass spectrometer is quite powerful, you can actually see the difference, and you can see this nice separation of peaks between them. Mass analyzers are usually used in tandem, so you don't use just one mass analyzer, you might have two or more mass analyzers coupled together. So this can provide you deeper insight into the molecular composition that you're breaking apart. So for example, if you just want one mass analyzer like we have on the first example, so you just let your sample go, so once it's ionized, goes to a quadrupole, it might be coupled with TOF, time of flight, and you separate your samples and they are detected and you are seeing peaks on the left. On the right, we actually have two mass analyzers in place, and in between them, you actually turn on a collision cell that fragments your ions into even more fragments. So you send your ions to quadrupole, then you send them to a collision cell. This fragments them even more, and then you send it to TOF, to time of flight mass analyzer, into the detector, and then you can actually see more information about particular ions. So not only that you are looking at that ion, but you fragmented it even more, so you can look more deeply into one. So we all often have these tandem MS or MSMS. So you can see here how it might work. So from ion source, we have an ion going into a mass analyzer, and we first analyze that, and we we get a spectrum of mixture, and let's say that we want to look at one peak specifically. So here on the left, you can see that we select one peak, we send that peak into a collision cell to fragment even more. And then we send those fragments into another mass analyzer, and we get spectrum of just that one peak that is selected. We match it against database that we want, and then we go back, we can pick another peak, we can fragment that peak, and we can analyze that peak. So slowly we can kind of build and look more into details through our molecule. So Peptides and glycans, they will also fragment in predictable location throughout their structure. So then we can, once we have all the peaks, we can elucidate their structure using MSMS in the database. So types of MS that are used in biopharma would be ESI-QTOF, so electrospin ionization sort coupled with quadrupole and then time of flight mass analyzer, and MALDI-QTOF, so matrix-assisted laser desorption ionization that is then coupled with quadrupole and then time of flight mass analyzer. Once you get your mass spectra, those mass spectra can be, you can compare them to databases. So you can use different databases such as MASCOT or MSFit or Profound, and you can compare your experimental data to theoretical data, and you can get, if you get a match, you can identify what you actually have. So what kind of protein you have, what fragments you have, what are the things that you're actually looking at. So the last thing that we're going to discuss are the application of mass spec for biopharmaceutical analysis. So our ICHQ6B that outlines different kind of specification and test procedures for our biologics and biotechnological products. This outlines the requirements for the analytical characterization of therapeutic proteins using mass spectrometry. And it does outline that LCMS is a powerful technique for the analysis of critical quality attributes. So it kind of like tells us that it needs to be, that something like mass spectrometry needs to be used during characterization of our drugs, because it's powerful. It can give us a lot of information with with confidence. Usually the way these analyses are done are with peptide-centric methods or bottom-up methods. So usually how our proteins are characterized is by chopping them up into smaller pieces and then looking at them using LCMS. So quite often you can see this peptide mapping being done in order to characterize your drug. So your protein sample is chopped up into peptides using an enzyme such as trypsin. So trypsin cuts peptide bonds in predictable ways. So from a protein we get short peptides. Those peptides can then be separated on using HPLC, and then, for example, we can use reverse phase peptide separation, reverse phase HPLC, and then we can couple that with mass spec and get accurate mass measurement and then determination of charge, and then we can do different MS coupling to get a lot of information from those peptides, and then through bioinformatic analysis we can determine actually what each one of those peptides contain. And then if you have any modifications on your peptides, you can easily see that. So once you know what your drug should be, and then you can compare it with a new batch of drug, and if you have any modification, you should be able to see it. So if something like deamidation happens, that will cause change in mass. So you will get a different peak. So you will know that something is different with your peptide. Your N-glycosylation, if it changes, again, that will change mass. If oxidation happens to one of your amino acids, such as methionine, that will increase your mass by 15.99, so that will be seen as well. So using mass spec, you can really see small differences on your proteins, so such as different changes even on one amino acid side. And this is one of the examples just to show you how mass spec and HPLC can be coupled to give you quite a lot of information about your drug. It is used quite a lot now when comparing innovator drugs with biosimilar drugs. So a lot of biopharmaceuticals are coming off patent now, and many different companies are trying to make copies of these big blockbuster drugs. So these copies are called biosimilars, and now mass spec is really used to compare biosimilars to innovators, because in order to get biosimilar to the market, you need to show that your biosimilar is as similar as possible to an innovator. So what we are seeing here is comparison of trastuzumab, which is an original drug with biosimilar using mass spec. So intact mass analysis, what we are seeing here in green is innovator and blue is biosimilar. So mass spec of intact molecule, and we see different heavy chains with different glycans uh, attached to it, so different sugars attached to it. So we have G0, G0F, G1, G1F. So these are just different heavy chains of a drug with different sugars attached. And you can see that they're comparable to biosimilars. We have all the same peaks, but those peaks are slightly shifted. So this analysis revealed that there was 32 kilodalton difference in each peak of the heavy chain in biosimilar. So basically, biosimilar was 32 kilodalton lighter than innovator. So that means that there is some difference in the structure of biosimilar. So when they looked at it in a little bit more detail, so they fragmented these heavy chains a bit more by using further MS analysis, so coupling mass analyzer. These differences are revealed to be due to amino acid substitution. So wherever innovator had glutamate, biosimilar had aspartate, and wherever innovator had methionine, in biosimilar there was leucine. So there was actually different amino acid sequence, which caused slightly different amino acid sequence, and that caused difference in the weight of one drug compared to another. Then they did a bit more analysis using mass spec and HPLC to look at the glycans, so to look at different sugars that are attached to this drug, and they show that there are actually identical glycoforms between both therapeutics, and difference was only in relative abundance. So if you compare innovator on top and biosimilar, you can see that we have all the same peaks in same position. So that means that all the same sugars are present, and the sugars are listed there on the right. But there was just slight difference in their abundance. So you can see, for example, there was a bit more of G1FA in innovator than in biosimilar. So you can see that by the size of the peak. And then another thing that they looked at modifications on amino acids. So on an amino acid level, they have seen that innovator and biosimilar actually have slightly different amounts of oxidized amino acids present. So in innovator, the oxidized species present only had percentage area of 4.95, while biosimilar there was a bit more, so there was around 11.29% of oxidized amino acids. So there was slight difference in these kind of modifications as well, which is again, you are able to see that using something like mass spec and HPLC. So this is a good way to compare innovator and biosimilar, and many companies are doing that. In this case, you could see that there was quite a lot of differences between biosimilar and innovator. So this wouldn't be a drug that can be a potential candidate. But it's good by doing something like this quickly, you can rule out that drug and say this one shouldn't go into further analysis, or maybe we have to do some modification in order to get it more similar to an innovator. So quite powerful techniques. Okay, so this is all for today. I think there are some more information on your Moodle page, so you can have a look at that. And let me know if you have any questions for now. Naomi is writing. Now he's asking, is the last exam we have online or in a test center? So your last exam or long answer question exam is going to be online as well. The only one that you have in NIBRT is the one on the second day of your practical. We have a few more questions. We'll just wait. Katya is asking about sample of the long answer questions. Yeah, I'm going to give some in the last lecture next week. So at the end of the lecture, you will get some samples of the long answer questions. And Rita has a question about the assessment in NIBRT. Yeah. It's going to be the questions related to the lab session or what we have learned so far. So your assessment in NIBRT, there's only going to be questions that are related to your lab sessions that you're going to do in NIBRT. No problem. Do you guys have any other questions for now? Rita is asking, will we need to gown up there? We are only going to be in lab, so you will only need the lab coat and safety glasses and gloves. So we will provide that for you. You will not need to gown up any further than that. Yeah, makeup is allowed.